Question mark, question mark. Yeah, my reaction too. I have a technology curse, in case you didn't know. It's where technology doesn't work for me because I'm me. All right, so first of all, there is a test for you guys. It is on the 9th. That is 1 week from today. There's a study guide on Canvas in the study guides module right there, which is right below lab documents. How would you have known? Well, you could know by reading the syllabus, which I'm 100% certain all of you did. You need to know what will happen on each class day because I have written it all down for you on the syllabus, the very last page. All right, before we start, you need to either have your lab handout or a PDF of the lab, which I mentioned in the announcement, so you should already have that. And at this point, you need to download the pictures of the results, which is what you'd see in lab if you did the experiment. Right under lab documents, I have put a new module entitled Lab 2, Cellular Building Blocks. This is what you would see if we were doing the lab. So download the results and download the questions page. That's where you're going to type your answers, and then you're going to submit that document here. Okay, so go ahead and do that. I will wait about 3 minutes. Let's find a timer. I'll wait 3 minutes, and then we'll start lecture again. Dismiss, dismiss. Okay. It has been 3 minutes. I will assume that you have downloaded. Let's see if there's anything in the chat. Why is it recording my words? That's weird. There's the chat. Okay, cool. So let's continue on. I'm assuming you've downloaded these things. I'm going to give you the pre-lab lecture, and then you're going to go ahead and analyze the results as if you had done them in lab. Our primary objective today is to identify the macromolecules, the reducing sugars, starch, triglycerides, and proteins. Those are the ones that we talked about in chapter 3, and we're going to identify those in common foods. Predict if the 5 common foods will contain starch, protein, reducing sugar, and lipids. And then you're going to test, well, you would have tested, but you're not going to test. So today's foods we're going to use are dog food, protein shake, Jell-O, potato chips, and Splenda. Now it's time for you to read pages 8 through 10 of your lab handout carefully. If you have the lab handout in front of you, you can read that. Otherwise, you will need to go to Canvas and under Lab Documents, download the Principles of Biology Student Lab Manual and read that. And we will reconvene. This little notification is for me to start the online timer. So I'm going to go to the online timer and start that. And we will begin in 9 minutes and 54 seconds. So this time you should be using to read pages 8 through 10 of your lab manual. Okay, that concludes our 10 minutes. So let's go back to the PowerPoint. And first two molecules or terms that you're going to need to know are reagent and reactant. A reagent is a solution used to test for the presence of specific organic molecules. We're testing for things using the reagent. And then the reactant is whatever you're testing for. The reactant reacts to the reagent. This I want you to write down. Reagent equals indicator solution and reactant equals macromolecule. You will need that for one of the post-lab questions. So I'll set the timer for, let's say, 30 seconds. I don't like using the timer this much, but it really helps me when I can't like see people engage their progress. Be like, okay, the class is done now. Right. So these are the reagents, the things that we are testing with. We have the Benedict solution, which tests for glucose, the iodine solution, which tests for starch, the Sudan 4 solution, which tests for fats and oils, and the Biuret solution, which tests for protein. Now, thank you, Mother Nature. We will not actually be using these, but this is what you would be using. And then we have 4 known reactants or macromolecules. We have starch, glucose, mineral oil, and protein. What you're going to do are make controls, and that will show you if the reagent or if the reactant is present in a solution. So if you combine iodine with a solution that you know has starch in it, then you're going to get this result. And if you combine iodine with water that has no starch in it, you're going to get this result. So this is a positive control. It shows you what it looks like if the result is positive. And this is a negative control, and it shows you what it looks like if the result is negative. We're going to pair these up to prepare positive controls. What you're going to do, well, you won't do it, but you would make a positive control, which shows what it looks like if the reactant is present. And question number 1, I think it's on page 12 of your handout. And if you're using the typed up version, I guess it's on the first page. The positive control is made by taking a solution of the known macromolecule and adding a reagent. And that is the answer to the first part of number 1 on page 12. You will observe a color change if the macromolecule is present. So for example, starch and iodine. Iodine is the indicator solution, the reactant, and starch is going to give you this. A negative control is the exact same, except it shows you what it looks like if the reactant is not present. That we use the reagent plus the water, and that's the answer to the second part of question number 1. You will not see a color change. So if you combine water and iodine, it's going to look this clear amber color. And then you can test for all the other foods and visually compare them to the negative control and the positive control to see if your result is negative or if your result is positive. You're going to use the positive and negative controls to determine if the macromolecule is present. Remember, the macromolecules we're testing for are sugars, starches, lipids, and proteins. Here's an example of what you might see if you were in the lab. So we have the iodine test for starch. Over here we have the negative control, so that's what it looks like if it's negative. And then over here we have the positive control. That's what it looks like if it's positive. And you can see that the experimental vial matches the negative controls. So we know that it is negative. It has no starch in it. This one is kind of cloudy, but it definitely more matches the positive control than the negative control. And you would get results like this in real life because nothing is perfect. But because it more matches the positive control, we say it's positive. The next test is for reducing sugars. A reducing sugar is a sugar that gives an electron to another molecule, like glucose is a reducing sugar. So here we have the negative control. That's what it looks like if it's negative. And the positive control, that's what it looks like if it's positive. And this guy is negative because it matches the negative control. So there is no reducing sugars in there. In this example, we have another food, and it is called weakly positive because it doesn't match the negative or the positive, but it matches more closely the positive. You can see the cloudiness going on in there. And food number 4 over here is more positive than it is negative, so we say it's positive. When you test for lipids, you're going to look for two things. You're going to look for these fat spheres, because fat kind of forms bubbles, or you're going to look for a separation. So remember how oil and water don't mix, because oil is nonpolar and water is polar. So anything with oil in it is going to separate and form this layer on top of the water solution. So if you see that separation, you know it's positive for lipids. And there's a question on page 12, which you can't see on my computer screen, but it's number 4. And it says, what about lipids causes them to react this way? What about lipids causes this separation? So you'll have to answer that. The next test for proteins involves a very subtle color change. So if you were in the lab, it would be helpful to use a white paper as a background. The positive control is this sort of darker violet color, and the negative control is this light blue. And this food in the middle here matches the positive control, so we know that it's positive. It has protein in it. All right, let's do the first part together. I hope you have this in front of you. Let's do the test for iodine. So first we're going to write the name of our test solution. I'm going to show you how this is done, and then you're going to do the rest of the tables on your own using the results that you downloaded. Iodine test for starch. So our first food is going to be dog food. So you're going to write that in line number 3. Then we're going to have the protein shake, Jell-O, potato chips, and Splenda. Now, the negative control we know is going to be negative. So you write a negative under prediction. And we know the negative control does not have starch. The positive control, you know, will have starch. So it will have a positive result. So you put positive there. Is everyone with me? Am I going too quickly? Let me check the chat. Do I need to? It's so hard to gauge like where the class is and how you guys are doing. Yes, I'm going too fast, or yes, okay. Am I going too fast? Yes or no? Oh shit, I am going too fast. Okay. What slide do we need to go back to? I guess you wouldn't know because you don't have the slides in front of you, so yay. Let's go back to the control. See what the chat says. Table 1, okay. Let's see what someone else says in the chat. Yeah, okay, so table 1. Where is table 1? Yeah, this is table 1 right here. So you're gonna write dog food, protein shake, Jell-O, potato chips, and Splenda. Maybe I need to give you some time to write that down. Let's start with 30 seconds, or maybe 1 minute. I have no idea how fast people write when I can't watch them write. All right, there we go. So the negative control is just iodine plus water. We know there's no starch in the negative control. So prediction, yeah, we're predicting a negative result. The positive control, we use iodine plus starch, something we know has starch in it. And we're predicting a positive result because that's our positive control. Then we have to guess for these guys. So we're talking about starch here, dog food. Well, I've had dogs in the past, and I know that a lot of dog food has a lot of grain in it, and grain contains starch. So I'm going to predict positive for starch. I'm going to put a plus right there under the prediction. Protein shake. Well, that's protein. It's probably not going to have starch, so I'm going to put a negative there. Jello. I can't imagine that would have starch in it. So that's a negative. Potato chips. Potato chips are like potatoes are purely starch. Like starch, fat, and salt, all the good things in the world. So I'm going to put a positive prediction there. And Splenda, well, Splenda has like nothing in it, so I'm going to put a negative result for that. All right, so does everyone see how I did that? I looked at what it was being tested for. Iodine test for starch. And then I predicted, based on my knowledge, whether I was going to see a positive result or a negative result. All right. So if you downloaded, which I hope you did, the results, this is what you're going to see. We have our negative control right there, and this is what you would use in the lab to make it if you were making it, but you're not, so you can ignore that part. The negative control looks like this, just iodine plus water, and the positive control looks like this, iodine plus starch. So what are we going to do? Oh, yes. So let's take a look at this. Well, for the negative control, obviously it's negative. So I'm going to write negative right here in my table. The positive control obviously gives us a positive result. So I'm going to write positive right there. Now, dog food. Let's look at the dog food. That matches the positive control. So that is a positive result. So over here, I'm going to write positive. Next one, protein shake. Protein shake. Oh, that's positive too. Protein shakes have starch in them. Well, that's not very healthy. Okay. Starch is not a health food. Interesting. I imagine that if we had looked at some nutrition facts, we could have guessed that. And in fact, in the lab, you would have looked at nutrition facts. But anyway, that's a positive result too. Okay, number 5, Jello. This one matches the negative control. So we know that it is a negative result. So I write a minus there. Potato chips. That one matches the positive control. So I write a plus. No, I keep pressing the wrong arrow. I write a plus right there. And number 7, Splenda. This matches the negative control. So I write a negative. So there is no starch in Splenda. Now you're going to repeat this process for tables 2 through 5. I have crossed out some of the comprehension questions in the typed document. You do not need to answer those. When you're done, you submit it right here. You submit the document, or if you are using your lab handout, you will submit pictures of your lab handout. And that is the end. Are there any questions? If you have questions, I will stay online. This should not take you more than 30 minutes. So I'm going to stay online, I guess, until the last person logs out. And if it takes you more than 30 minutes, that's fine. Someone's typing. Could you go back to the slide that explained number 4 on page 12, please? Yes, I can do that. Click to exit. Okay. Number 4 on page 12. Was that? No, that's number 1 on page 12. Wait, which one is number 4 on page 12? Which slide is it? There we go. Okay, so the question is, no, come on. What about lipids causes them to form a separate layer on top of the water solution? There's a specific word for it, and it's a word that I spent a lot of time on in chapter 2. It has to do with things being polar or nonpolar. So think about that for a while. You can go back through the chapter 2 PowerPoint if you need to find out why lipids and water do not mix. But you can't just say oil and water don't mix. Everybody knows that. Go a little deeper. What is the name of the chemical property that causes oil and water not to mix? So we just need to fill out the remaining 2 tables on page 11 and submit it. Let me find my lab handout. It's floating around here somewhere. Man, I used it yesterday to make the PowerPoint and then I didn't put it away. Page 11. Let's take a look at the lab handout so that I can see exactly what you're talking about. All right. You need to fill out pages 11 and 12. If you are using your own lab handout, fill out pages 11 and 12 and answer all the questions except number 3 you can skip. Number 3 on page 12 you can skip. Awesome. Any other questions? I will stay online for you to ask me questions when you have them. And whenever you're done, just submit and log off.